next speaker is Dr. Mathew John. He's from Trivandrum, and his topic is going to be on endocrinology of aging. And there is a magic potion ahead. We all need a magic potion, I think, at this at this age. So, dais is for you now.
Yeah, thank you, sir, for the introduction. Special thanks to Dr. Krishna Sheshadri and team for inviting me here. So basically, I'll be looking at the endocrinology of aging, and is there a magic potion ahead? Conflicts of interest, none. Unlike Krishnan Swaminathan, I have not done any major research in aging other than looking out for hair dyes. Bijan seems to be the best. So how old are these guys? I think the question is not very relevant because most of us in South India know these people. This is Mammootty, who is 68 years plus. So, and the other guy on the right side is in fact a random person whom I searched from the net asking for a routine 60-year-old man, and this is what you see. So these are certain external manifestations of aging. So what are the sort of concepts of aging now, and how is the endocrine system associated with all this? Now, aging is a major risk factor for various comorbidities, which includes CKD, dementia, diabetes, cancer. When we talk to patients about aging of pancreas, we always say this: you are like something like a car, and as years go by, you need to replace many of your parts. Some of the things can get worn and torn. So similarly, almost all the organs in the body can have some sort of wear and tear as the aging happens. But the question also remains that, are these essential parts of aging? In fact, no. There are people who can age to, say, 100 years without any cardiovascular disease or dementia or cancer. And there are some people who can get all this disease at a very early age. And we are all aware of the fact that the population, as the population ages, the disease, chronic disease can increase. This is the sort of typical population pyramid that you would see on the left side, and on the right side, what you would see is how the population pyramid has changed over a period of many years because of an increasing number of aging population. Coming to alterations of the aging body, there is reduction in bone, skin, and skeletal muscle strength and mass. There is derangement of insulin signaling, derailing on to various problems of diabetes or glucose intolerance. Adipose tissue can increase, the fat mass can increase. It can worsen the insulin resistance. There are various changes in the immune function and changes in the endocrine gland functions and end organ sensitivity. So primarily, I'll be looking at the endocrine part and then touch a bit on the magic portion of it. So I'll just run into individual things beginning. It's glucose regulation in aging. We are all aware that as you age, the glucose intolerance increases, and more than 50% of people more than 80 years of age have, in fact, some form of glucose intolerance. What is the reason for this? Not very clear, but one is there's a relative reduction in the insulin secretion by the beta cells in comparison to the insulin resistance. And there is increase in peripheral insulin resistance due to poor diet, chronic illness, physical inactivity, increased abdominal fat mass, and reduced lean body mass. In studies which looked at whether chronological age is an independent risk factor for glucose intolerance, less than 10% of the variance could be explained by age, and the rest of it was related to age-related changes in fat mass or body mass or lean body mass, which accounted for glucose intolerance. Thyroid changes in aging is again very well known. We are all aware that the TSH levels of treatment or TSH levels of targets are revised for people who are very elderly compared to that who are younger persons. The TFT, if you look at values of patients who are, thyroid function values of patients who are elderly, you will see that there's an increased risk of subclinical hypothyroidism. And also you see people with a very extended TSH range, some people with very low end of TSH, with suppressed TSH, mostly related to increased risk of nodular goiter and toxicity in patients who are elderly. The general thyroid function changes of patients who have been longitudinally followed up has been shown something like a sick euthyroid syndrome. You tend to have a slow reduction in your T3 levels, a free T4 levels which are normally preserved, and TSH levels which are normal or slightly on the lower side. Physiological TSH increase in aging is related to the, possibly related to the lack of the pituitary sensitivity to circulating T3 and T4. Subclinical hypothyroidism is quite common. Autoantibodies are quite common in elderly, but we are aware of the reason why we should not be treating these patients very aggressively. Similarly, both patients with hypo and hypothyroidism can have atypical presentation as the people age. Menopause is probably the most dramatic change in hormones which happens in aging. The real reason why it's partly believed to be partly due to a reduction in ovarian reserve or partly due to a change in hypothalamic neurotransmitters is still being debated. And changes similar to aging has been seen in younger patients who are going to premature menopause, which includes osteoporosis, urodegenerative atrophy, and mood changes. But various studies trying to replace or recreate the normal internal milieu or the hormonal milieu of premenopausal women in postmenopausal women has never been found to be really successful in terms of outcomes like hard outcomes like cardiovascular or bone or stuff like that. Coming to andropause, which is probably a less dramatic event, starting from the age of 40 years, you have a gradual reduction in both total and free testosterone happening. Unlike menopause, it is quite gradual. And different reasons are there, but the most common reason postulated is the impaired ability of Leydig cells to respond to gonadotropin. And 10 to 25% of elderly men have a testosterone values less than 3 nanogram per ml. This is accepted lower limit of normal for a young adult. But not all men, elderly men with testosterone less than 3 nanogram per ml are symptomatic, and only symptomatic men with testosterone less than 3 nanogram per ml should be replaced. And there's a 2015 ADA and FDA regulation that only elderly with a function or with an organic cause of hypogonadism should be replaced with testosterone. If you look at the overall evidence of testosterone replacement in elderly people, even with those with normal testosterone or low normal testosterone, there has been minimal to no improvements in sexual dysfunction, physical function, or cognitive function. Improvements in bone density, improvements in lean body mass, and grip strength has been seen. So for each individual patient that you see when you plan to replace testosterone, look at the risk-benefit ratio before you start replacing it. The next organ, the endocrine organ which can start aging is the adrenopause. The adrenal glands can get aged. And the DHEAS levels can have a slow decline in people beyond 20 to 30 years of age. And by 80 years of age, you almost reach a pre-adrenarchal levels of DHEAS, which is almost like a normal DHEAS levels for a child. There are certain associations of DHEAS levels with diseases like Alzheimer's and cognitive dysfunction. But looking at trials, various trials with DHEAS replacement has been attempted. You can see that none of them has produced any dramatic benefit for men. In women, certain minor improvements in terms of libido and sexual dysfunction has been improved by DHEAS, but overall benefit in elderly DHEAS replacements has been very minimum. The next axis to get affected is the GH-IGF-1 axis. Somatopause, again, a very gradual, not very consistent decline in GH production, and IGF-1 levels happen with aging, start from the age of 30. Again, replacement of growth hormone in elderly with lower IGF-1 levels has never been found to have any major benefit in terms of long-term outcomes. And GH replacement in elderly has been associated with problems like carpal tunnel syndrome, edema, arthralgias, and glucose intolerance. So if you look at all the hormone replacement therapies that we just learned about, be it adrenal replacement, be it GH replacement, testosterone, or postmenopausal hormone replacement therapy, none of these replacement therapies have been found to recreate a normal youth in people who are aged. So rather, most of these changes in elderly in terms of endocrine organs seems to be a part of normal aging, and I don't think any such replacements are going to show any benefit in the future. Now coming to certain concepts in aging, I think when we say aging, we are all looking at people who you would like to prolong your life from, say, 75 or 76, which is the normal expectancy of life, to, say, 100 or 110. That is the lifespan of an individual. But when you prolong the lifespan of an individual, you also prolong the time which this person is going to live with a chronic disease, be it a cardiovascular disease, cancers, or Alzheimer's. So you prolong the entire lifespan. So now the concept is that instead of prolonging the entire lifespan, what you're going to do is to prolong the health span. So health span is by definition is the time or a period of your life when you are living free of diseases. And you're trying to reduce the morbidity part to a very small part of life, saying that you live for 110 years and you live with a chronic disease for only 5 years, the last 5 years of your life. So extending the health span and compressing the comorbidity is what people are into research now. And a new branch of gerontology, or a new branch which is developing, is called a geroscience, which actually tries, just like you would see in this picture, to change the incidence of comorbidity from an earlier age to a later age, is what they are trying to essentially do. And this science targets certain biological aging processes to delay or prevent the incidence of chronic diseases which can occur during aging. And there are various trials which are designed to look at this. Now, aging is a very complex process. It's not a single process which is happening. So to look at a trial of aging, you need biomarkers. And you can't have a single biomarker. And the biomarker should definitely defeat age as a biomarker if you're looking at aging as a process. So there are various processes which constitutes aging. And I just put some of them here, that example, the telomere attrition, the genomic instability, loss of proteostasis, cellular senescence, and various other factors which are pathogenesis of aging. So if you look at biomarkers, one single biomarker cannot explain this various pathogenesis process. And you need to have different biomarkers to be taken in total to look at aging. I just look at two biomarkers before I proceed further. One is telomere shortening and aging. It's probably one of the most closely looked at biomarkers. Just to revise a bit of genetics, I too just read it up yesterday. So telomeres are actually small caps at the end of the DNA. And you have got a double-stranded DNA. So each DNA will have each of the strands will have two caps. It is just like you putting the cap on a pen. You have the pen and the cap just to protect the nib from getting damaged. Similarly, the telomeres protect the DNA from getting damaged. So as the cells divide, these telomeres get shortened. Some of the telomeres will get eaten up during the process. And once the telomere length reaches a critical stage, then the cell cannot divide anymore. The cell goes into senescence. So telomeres act as an aging clock in the cell. And if you look at telomeres, you can understand how much the cell has aged. The germ cells and certain cancer cells have got telomerase, which actually will repair this fast so that the cells keep on repeatedly replicating. And that is one of the reasons why telomerase inhibitors have been looked at in cancer therapy. So telomere is one of those markers. Second is a new term again, which I learned it's called proteostasis. So proteostasis is something like homeostasis. And it basically means that you're trying to preserve a protein in its original form. When you get aged, all your proteins also get aged. So protein is going through a mechanism of rebuilding and again getting destroyed. And this happens through ribosomes, which make the protein, the chaperones, like the heat shock proteins, which actually gives the tertiary and quaternary conformation to the proteins. And there is a protein degrading mechanism. So as you age, these proteins also get aged. And protein aging is believed to be one of those mechanisms leading to aging. And failure of these mechanisms can, in fact, lead on to aging. And certain proteostasis agents have been looked at biomarkers. I'm not going to explain this slide, but it just shows a list of biomarkers in the blood, which are used in various clinical trials to look at aging and to study aging over a period of time. Now, the last part of my talk, is there a magic potion ahead? Basically, calorie restriction and longevity is one of those earliest studied mechanisms of aging reduction or prevention of aging. And studies, especially in various primates, including rhesus monkeys, have shown that group of monkeys exposed to calorie restriction, in fact, lived longer than monkeys who were having an ad libidem intake of calories. And there are classic studies, one of this which I'm just showing here. I'm not going into any further details, but just to tell you that calorie restriction has produced various benefits in terms of animal models and even human studies, but none of them really related to measuring the life of a person. But it produces preservation of cognition, delays sarcopenia, protects colonic health, delays brain atrophy. So various things which has been shown by caloric restriction. And the mechanism of caloric restriction also has been found out to act through various pathways, which includes AMPK, mTOR, PPAR, sirtuins, etc. The importance is that it's very difficult to practice calorie restriction in real life. But the important part is that studying calorie restrictions has led on to various pathogen mechanisms being found out. And we now have molecules like metformin, which acts on AMPK, resveratrol, which acts on sirtuins, rapamycin, which acts on mTOR. all this has been found to delay aging. And this is one of those classic trials in human beings, the calorie trial. It is the very new calorie, the part of the calorie trial is being discussed much more than the original calorie trial. This is the original part of the calorie trial, which looked at 25% calorie restriction in people 21 to 50 years old with a normal BMI. And what they looked at is two outcomes: reduction in resting metabolic rate and reduction in core body temperature. And why they looked at these two uninteresting sort of stuff is that because in animal models, these two things have been found to prolong longevity in animals. So what they found is that at the end of 24 months and at the end of 12 months, there was an improvement in total body temperature, in total energy expenditure, but there is no change in core temperature. So some insights into how calorie restriction can help in terms of aging. There are various metformin studies which are going on. One is called the TAME, one is called the VA Impact. So different studies trying to focus on metformin, whether it can delay aging, and we'll have to wait for a few more years before we get results of the study. And another area which is being looked at very closely is the nutritional supplementation and aging, because there are at least some data to show that certain diseases like Alzheimer's, certain diseases like age-related macular degeneration, cataracts, diabetes, etc., can be prevented with certain nutrition. But we still don't know what is a sort of threshold that we are more which these patients get benefited by the supplementation of these drugs. So finally, to end my talk, just to summarize the entire thing, there are various pathogenetic mechanisms which constitutes the aging process. It's not a single mechanism. And the aging phenotype also differs, which makes it a very difficult problem to study. You can't call aging as a disease to study, it's a problem to study, because you can see people who have certain problems like significantly reduced bone mass, but they don't have a cardiovascular disease. Certain person will have frailty, but they don't have any other illness. So it's a very difficult phenotype to study. But we are in the process. We are in the process of good aging research, which is happening world over in terms of genetic manipulations, in terms of drugs, in terms of dietary changes, in terms of exercise. So I end my talk here. Thank you very much.
So thank you so much, Dr. Mathew. If you're not rushing, I thought we'll finish the next session.
No, I'm rushing.
You're rushing. Okay. Then we'll take the questions now. Any questions from the audience? So before any questions come up, there used to be a lot of talk about studies done in yeasts and nematodes in the past by mutations of the insulin and IGF-1 signaling pathways. And that could prolong the lifespan of these organisms, and then that could be replicated in animals and humans. Is that something that is happening, or?
Yeah, like insulin growth factor 1 related thing has been a point of interest. But overall human data is not very encouraging, because even epidemiological studies have shown it seems to be sort of U-shaped relationship. People who have very low IGF-1s and people who have high IGF-1 tend to have an increased mortality. There's somewhere a balance in between which keeps sort of optimum lifespan. And most of this animal, mostly in nematodes, most of the studies have been taken in nematodes. But they are using these studies as a sort of backup postulates for human studies. But I'm not still aware whether anything concrete has come from the amount of literature I reviewed.
I couldn't find either, so that's why I was asking. So any questions from the audience?
This calorie trial, which I had just shown, there is actually a clinical component that's now there to it, and that has shown that this sort of calorie restriction can reduce the risk of chronic diseases, like cardiovascular disease and stuff like that. So this is a very mechanistic part which I just showed you, but it's got an entire, maybe I think at least 100 publications have come out of the entire calorie group of studies.
I think, thank you so much. We've come to the end of the session.